Greetings to everybody watching. I take this platform to discuss the topic Biophysics of Electrical Encephalography or EEG. So while we discuss the traditional views on EEG sources in the brain, we consider that activity in neural circuits produces the various EEG features linked to cognition. The standard model of EEG states that simultaneous postsynaptic potentials of neural populations produce EEG. But this explains only the existence of EEG and not the meaning of the content of EEG signal that we see in various ERP studies. Hence, no grand unified theories are presented because there is unlikely to be a single neural correlate of the whole EEG. So while we search for the real source of electrical activity in the brain, we can consider pyramidal cells to be one of the eligible candidates. Apical dendrites of pyramidal cells in bulk, say around 1 million synapses, are needed to active simultaneously in synchrony by creating a separation of both charges, negative and positive, on one of the either ends to create electrical fields around the brain that we may discuss in the further presentation, which is the real source of electrical activity that is captured by electroencephalography or EEG. As we can see in the green colored figure, there's a little difference between generic and pyramidal neurons, as can be seen here, as well as there is a little difference between action potential and postsynaptic potentials, which are signals within the neurons and between the neurons respectively. The process that follows for production of an EEG or generation of an EEG activity is as follows. When neurons are activated, local currents or electrical currents are produced. EEG measures the current that flows during the excitation of the dendrites of many pyramidal neurons in the cerebral cortex. Potential differences are caused by some postsynaptic potentials from pyramidal cells that create dipoles between soma and apical dendrites. Now, what are dipoles? Let's discuss them here. Dipoles are defined as a separation of charge over a distance. These are fundamental to measuring and recording activity using EEG. Neurons undergo action potential producing postsynaptic potential, as we all might be knowing. Well, here's a little diagram depicting the action potential that depicts the different processes involved in one single action potential spike, starting from depolarization, then repolarization, coming back to hyperpolarization, and then coming back to the normal resting membrane potential. This undergoes a neuron undergoes this whole process to create two types of postsynaptic potentials on the postsynaptic neuron. One is excitatory in nature called EPSP, excitatory postsynaptic potential, or another is inhibitory in nature called IPSP, inhibitory postsynaptic potential. Despite whether they lead to action potential or not, both of these postsynaptic potentials generate a separation of charge, which causes a dipole. So we just missed a point that for an EEG activity to be generated, there are two main conditions. One is alignment of neurons in a particular direction, and second, their synchronous activity. So by that we mean, as can be seen in the diagram above, the skull diagram, there are green colored pyramidal neurons here. Now these are aligned in a longitudinal direction, and there are also neurons that are aligned in a transverse or horizontal direction in cerebral cortex. So populations of these neurons in a particular direction, say horizontal or longitudinal, have to fire together in bulk, in abundance, in order to create a single EEG signal. Current dipoles, as we understand them, are pyramidal cell assemblies arranged with dendrites perpendicular to the cortical surface. EPSP, excitatory postsynaptic potential, at a postsynaptic pyramidal cell causes an influx of positive ions and depolarization of the cell, thus creating a current sink locally and current source at distant basal dendrites. These are the two cases of excitatory synapse receiving signal from excitatory synapse, and another example of receiving a signal from inhibitory synapse, where a positive and negative charge accumulates at either of the two ends. Intracellular current between source and sink constitutes the primary currents. The laws of current conservation causes currents to flow externally out of the neuron to generate volume currents, which form a closed current. This is the basis of this diagram here. Volume currents affect both EEG and MEG, but EEG more than MEG, and this is the basis of the EEG activity that is being captured by the EEG electrodes. This depicts current signals or current flow in the head compared to a cell battery. And these are the frequencies of electrical activity that is produced in the human brain, in form of which the EEG device collects the EEG signals from the brain. And here we end the presentation of this topic, biophysics of electrical activity, with the thought that pyramidal cells and the activity within them produces the real EEG activity. These are my references. Thank you.